Now, before moving further, yes, you must compare between DNA polymerase in prokaryotes and eukaryotes. In prokaryotes, that is E. coli. I take example of E. coli. Yes, E. coli is a prokaryote, you all know. In this, DNA polymerase 1 is there, DNA polymerase 2 is there, DNA polymerase 3 is there, and this is DNA G protein. So DNA polymerase 1, 2 and 3 and DNA G protein. Now, yes, eukaryote, the polymerase are named as alpha, beta, gamma, epsilon, delta. Alpha, beta, gamma, epsilon, delta. These are eukaryotic polymerase. Okay. So now you will remember, first of all, prokaryote, we have discussed DNA polymerase 3. Synthesize, yes, I have discussed. DNA polymerase 3 synthesize complete leading strand and small fragments on lagging strand. Those small fragments are called Okazaki fragments. DNA polymerase 1 remove primer and fill the gap. No, somatic cell don't have telomerase. Okay. Now, DNA polymerase 2 counterpart are beta and gamma. DNA polymerase 3 counterpart are epsilon and delta. Now let me tell you that how you will remember. First of all, you will remember the prokaryotes. We have discussed prokaryotes. DNA polymerase 1 remove primer and fill the gap. Yes, DNA polymerase 3 I have discussed complete leading strand and small fragments on long strand. Okay, so you can remember. Yes, complete leading and Okazaki on lagging. DNA polymerase 2 is responsible for DNA repair. DNA polymerase 1 remove primer and fill the gap. Okay, so now you know. In prokaryotes, primer are synthesized by primase. What is primase? Primase is the name of enzymatic activity which is present in a protein. If I say primase, primase is the name of enzymatic activity which is present in a protein. So we found that in prokaryotes, DNA G protein has primase activity. So in prokaryotes, DNA G protein synthesize primer. DNA G protein synthesize primer. So question is asked, in prokaryotes primer is synthesized by, you will say DNA G protein. In prokaryotes primase activity is present in DNA G protein. In eukaryotes, alpha polymerase has primase activity, means it synthesizes primer. Alpha polymerase synthesize primer. Primer is RNA. Primer is not DNA, primer is RNA. So name of this polymerase is DNA polymerase, but it is synthesizing RNA. So don't get confused. Sometimes they try to confuse you that alpha polymerase is DNA polymerase and the primers are RNA. So can it remove that? So can it synthesize primer? Yes, it synthesizes primer. Alpha polymerase synthesize primer, that those are RNA. Clear? Now, in eukaryotes, mitochondria is there. In eukaryotes, mitochondria is there. In mitochondria, there is double-stranded circular DNA. In eukaryotes, there is double-stranded circular DNA. Okay. Yes, double-stranded circular DNA, and this mitochondria this double-stranded circular DNA is synthesized by gamma polymerase. So you can easily remember, yes, alpha, beta, gamma, epsilon, delta. Only one polymerase having gamma matlab mitochondria. Okay, so gamma mitochondrial DNA synthesis, beta, epsilon, delta, no DNA, yes, no M, so no mitochondrial DNA synthesis. Yes, epsilon, we found that in eukaryotes, leading and lagging strands are synthesized by different polymerase. We found that leading strand is synthesized by epsilon polymerase, LEADING, leading. Lagging strand is synthesized by delta polymerase, yeah, lagging. So you can easily remember by this mnemonic, leading. E looks like epsilon. Leading strand is synthesized by epsilon, lagging strand is synthesized by delta, gamma mitochondria, alpha for primer synthesis primase, beta for DNA repair. Beta is for DNA repair. Okay, got the point? Now, all DNA polymerase synthesize in 5 to 3 direction. Yes or no? Because you know synthesis always occur in 5 to 3 direction. So all polymerase, DNA polymerase have 5 to 3 polymerase activity. So all these polymerase have 5 to 3 polymerase activity. Okay, got the point? 1 point. Alpha polymerase, beta polymerase, gamma polymerase, epsilon, delta, DNA polymerase 1, 2, 3, all polymerase have 5 to 3 polymerase activity. Okay, 5 to 3 polymerase activity. Now, what is proofreading? Understand what is proofreading. Proofreading means, suppose I am doing any work. If I found anything wrong, then I will correct it. That is called proofreading. Let me explain in a simple word. Let me tell you in a simple word. I told you that's the reason I am telling you that telomere is present at a linear end of chromosome. Chromosomes are not present in prokaryotes. Okay, good question. Okay, yes, in prokaryotes there is no genome, there is no nucleus, there are no chromosomes. Okay, now what is proofreading? Now understand, DNA polymerase 3 synthesizing in this direction. If I talk about you in your exam, yes, when you attempt a question, before going to next question, suppose you marked a circle C. Now you are moving to next question, but before going to next question, your mind is little bit suspicious and you check your previous answer. Is it correct or not? Yes, it's a human nature. We do have, during our exam, we do proofreading. And if we find, oh no, by mistake I have marked the wrong answer, you will correct it. In the same way, DNA is doing DNA polymerase is just like, means what is being done by DNA polymerase? Let just suppose DNA polymerase synthesizing in this direction. Yes, you know that even if a single nucleotide is wrong, this may lead to mutation, point mutation, that may lead to cancer, that may lead to cell death. So when DNA synthesis is going on, this is very vital. That's why proofreading is there. Suppose DNA polymerase join one nucleotide. Yes, DNA polymerase join one nucleotide. After joining one nucleotide, it was about to join next. Before joining next, it will check it. If it is correct, then it will move to the next. But if it is not correct, then it will cut this nucleotide from this direction to this direction, from this end. So proofreading means remove proofreading means 3 to 5 exonuclease activity. Yes, 3 to 5 exonuclease activity. Okay, understood? 3 to 5 exonuclease activity means it will synthesize in this direction, and if it find that after joining it is wrong, it will correct it by removing it from 3 to 5 end. So proofreading means 3 to 5 exonuclease activity. Okay, so this is about proofreading, and we found, so simply you can write here proofreading. Yes, proofreading means 3 to 5 exonuclease activity. Have you understood this? Have you understood this? Proofreading means 3 to 5 exonuclease activity. All polymerase have proofreading except alpha and beta polymerase. Except alpha and beta polymerase. All polymerase have proofreading except alpha and beta polymerase. Okay. Third, yes. So if I ask you, beta polymerase do DNA repair? Beta polymerase no proofreading. Is it possible? Is it possible? Yes, it is possible. DNA repair is different from proofreading. DNA repair is different from proofreading. DNA repair is different from proofreading. Proofreading means if I commit mistake and I correct it, this is proofreading. If you commit mistake and I correct it, it is repair. Okay. So proofreading. Clear? One question. Not only on leading strand, lagging strand, you will find on lagging strand the last primer. On lagging strand, the last primer will be removed. Yes, lagging strand, the last primer will be removed. When this last primer will be removed, then the gap will be filled by no one, can be filled. The last primer is removed, so yeah, this last primer is removed by DNA polymerase 1, but the last primer is not filled by the DNA polymerase 1, so a small portion of this. Okay, so last means this end and this end, both ends are small portion of DNA is lost, both on leading strand and lagging strand. Yes, this is the fact. Okay. She will, yes, DNA polymerase, yes, DNA beta polymerase has no proofreading, and yes, it cause, sorry, it's confusing. Beta polymerase do DNA repair, yes or no? Yes. So is it possible that it has no proofreading and it can do DNA repair? Yes. DNA repair is different from proofreading. Proofreading means, yes, you committed mistake and you corrected yourself, that it is proofreading. DNA repair means I commit a mistake and you correct it, or you commit mistake, I correct it, that is DNA repair. So proofreading and repair, these two things are different. Okay. Now I say, so this is clear to you. Now I say that now you remember these two things. Third point is, so third point, if I say DNA polymerase 1, if I make this DNA polymerase 1 like this, having 3 subunit, DNA polymerase 1, all polymerase has 5 to 3 polymerase activity. Yes, 5 to 3 polymerase activity, DNA polymerase 1. Okay, all polymerase have 5 to 3 polymerase activity. All polymerase have proofreading except alpha and beta, and proofreading means 3 to 5 exonuclease activity. 3 to 5 exonuclease activity. Yes, proofreading means 3 to 5 exonuclease activity. I told you proofreading means, yes, 3 to 5 exonuclease activity. 3 to 5 exonuclease activity. Okay, so 3 to 5 and one activity also present in DNA polymerase 1. Can you point out one more activity? Five to 3 exonuclease activity, which was present for primer removal. I told you primer removal. Yes, remove primer. Very good. Primer removal, we were having 5 to 3 exonuclease activity. Yes, I told you. Yes, DNA polymerase 1 remove primer. Yes, DNA polymerase 1 remove primer in 5 to 3 exonuclease activity. 5 to 3 exonuclease activity. So now you can see that DNA polymerase 1 has 3 fragments. One is this activity, second is this activity. I have taken separate color for this. The reason is that we can delete this, means remove this part of DNA polymerase 1 enzyme, this subunit of DNA polymerase enzyme 1 enzyme. If I remove this unit, the remaining unit is called clean-out fragment. Clean-out fragment. This question was asked in NEET recently. What is clean-out fragment? Clean-out fragment is DNA polymerase 1 without 5 to 3 exonuclease activity. So why this cleaner fragment is formed? Why this cleaner fragment is formed? This cleaner fragment is formed because we require it in PCR. We required previously, yes, in PCR polymerase chain reaction, we used to use it for replication. Yes, in PCR we used to use it for replication. So PCR we used to use it for replication. So this is cleaner fragment. Okay, so this is question. So we can say DNA polymerase 1 has 3 enzymatic activity, DNA polymerase 2, 3 are having only 2 enzymatic activity, yes, 5 to 3 polymerase and 3 to 5 exonuclease activity. 3 to 5 exonuclease activity means proofreading. Okay, many time question PGI that what is exonuclease activity, 3 to 5 what is proofreading 3 to 5 exonuclease activity. Which of the following is not having proofreading? Alpha and beta polymerase. So now you can remember. Okay, now primer removal in prokaryotes. DNA polymerase 1 remove primer. DNA polymerase 1 remove primer and fill the gap, you all know. Okay, in eukaryotes primers are removed by nucleus RNAs. Yes, RNAs and FAN1. RNAs means it remove is RNA. FAN means flap endonuclease. FAN1. Okay, and delta polymerase. Yes, mitochondrial inheritance must be taught by Dr. Parveen, patho, flap endonuclease, delta polymerase. In mitochondria, RNAs, FAN1. Okay. Yes, there is a book, standard textbook, Devlin has mentioned that delta polymerase has a role in cutting this primer. Yes, primer is cut by delta polymerase. Okay, it means a small cut is done by delta polymerase. Then FAN1 flap, it makes flap, and then RNAs cut RNA and primer is removed. But in mitochondria, RNAs and FAN1. Okay, mitochondrial inheritance, you must have learned that this is ovum, this is sperm. Yes, in this sperm, yes, you will find that mitochondria is not going to the ovum. Yes, mitochondria not going to ovum. Yes, so this ovum mitochondrial DNA is inherited. Yes, mitochondrial DNA of ovum, mitochondrial DNA of ovum goes to next generation. Yes, mitochondrial DNA of ovum goes to next generation. So if a mother, father, yes, you will find that if mother is ill, yes, having illness, then all children, it may be male or female, would be affected. Okay. Now if this male having mitochondrial, yes, inheritance, yes, many things are discussed in patho, so it can spare our time. Yes, if this person yes, marry to female, then you will find the yes, the this is all children would be normal, but again mother married to a normal male, then this then all children again would be affected. Correct? Do you agree with this? So mitochondrial inheritance, so I don't discuss further, just have an idea you have discussed with patho. So hope you have understood till now. Are you comfortable with this? Have you understood? This is very very important. Again I say difficult, no doubt, but you can easily remember by mnemonic types, that leading, yes, leading, lagging, who will synthesize a small fragment. After each replication, small portion is left, not synthesized, lost, telomere. Cancer cell, telomerase is there, telomerase is not ribozyme, yes, important. Reverse transcriptase, yes. Somatic cell have Hayflick limit, recently asked question, yes. Yes, this comparison question can be asked, yes. All these enzymatic activity you know, yes, and you know that clean out fragment, recently asked question, so we can't skip. Mitochondrial inheritance, they can give you pedigree and they can ask. Now we discuss